Kia ora everyone. Welcome to session 2 on day 3 of the 2020 Forest Growers Research Conference. The session is titled Breeding and Biotechnology, and over the next 2 hours we have 8 of our colleagues presenting on topics covering a range of species and breeding techniques. My name's Shaf Van Bellicum. I'm the General Manager of Proceed, a Ngāi Tahu business and a major provider of germplasm to the Australasian forest industry. All of the topics in this session have direct implications for our business. Some of them I regard as disruptive and will significantly change the way Proceed does business in the next decade. Others may be regarded as orthodox or more business as usual, but also provide opportunities to add value to both our business and the wider forest industry. Our first speaker is Paul Watson. He's the CEO of the Radiata Pine Breeding Company. Paul's fairly new to this role, having been with the company for about a year. Prior to that, he spent 25 years managing a series of very large pulp and paper research programs in Canada before returning home to take up a position with OGI and latterly the RPBC. The next decade will prove exciting for the RPBC and our industry as well, as we build on our investments in both genomics and plant propagation to deliver the gains that genomic breeding promises, and we'll hear more about that in later presentations. Paul's plan today is to simply bring us up to speed with developments in the Radiata Pine breeding program space, including the genomics program itself, maybe new breeding analysis, changes to RPBC structure, and offering his thoughts about what is on the horizon for the company. Many of our key breeding and selection traits are the result of multi-gene effects. And our next speaker, Gancho Slavic, his research at Scion is seeking to understand what drives this expression, particularly in different environments and with different management techniques. Now, Gancho's topic today is very broad, but essentially he'll talk about how we're using genomics in the Radiata program, and believe it, because it's already started. In addition, he will offer views on how to use these technologies in other species. Now, Gancho has a PhD in forest genetics, and for a lot of his career he worked in the US with Glenn Howe and Steve Strauss in the areas of Douglas fir genomics and poplar genomics. So his experience is going to be invaluable in showing us how we can practically use these technologies in our breeding program in a cost-effective manner, because it's not cheap, but also how companies such as Proceed can use these technologies also in the management of our seed orchards. Okay, so we've got a process, a genomics process for providing new germplasm. How are we going to get this germplasm into our forests? Well, in 2019, a research program called Tissue Culture for 21st Century Forests started. Now, the program is focusing on a new approach on developing clones, or varietals, as we are calling them, of Radiata pine from a combination of somatic embryogenesis and genomic selection using bioreactors and robotics. And that'll become a little bit clearer in the presentation. The objective of this program is really a very cost-effective production of varietals, and this will lead to significant uptake by industry, we believe. Now, we have 2 presenters, Associate Professor Jana Kranikova and Dr. Russell Burton, who will take us through the key elements of the program. Jana is a research leader for the Tissue Culture Group at Scion, and prior to coming to New Zealand, has held several senior roles in molecular biology and genetics in universities in Slovakia, Italy, and Finland. Russell will be known to many of you. Prior to his taking over a role as program manager a year ago for this program, he was the general manager for research, investments, and multi-partnerships at Scion. Then we're going to jump species and look at the role of breeding to improve heartwood in dryland eucalypts. Now, this research program is directed by Dr. Clemens Altena at the School of Forestry. Now, Clemens is a leading wood scientist with a Doctor of Natural Sciences from the University of Hamburg. Now, as you know, natural durability within a timber species can be variable, and orthodox wood quality applied research is often expensive and relies on small numbers of samples. Clement's work is focusing on the efficient testing of thousands of genotypes at a young age for heartwood quality and quantity within Eucalyptus persistoana in the first instance. The results of his research are then being used to rank selections in the persistoana breeding program. After that, we're going to enter an area some call the dark arts, genetic manipulation. Our presenter for this presentation is Glenn Thorlby from SCIEN. Now, Glenn is a plant molecular biologist with lots of experience in gene discovery and functional genomics. He leads SCIEN's plant biotechnology research program. Glenn gained his PhD from the University of London and has been at SCIEN for the past 10 years. And while the title of Glenn's presentation initially was to focus on ideas such as sterility, he will in fact talk about the broader suite of molecular technologies and the opportunities arising from these. Transgenics and gene editing are ones that many of us are familiar with, but there are others, and rather than modifying or adding to plant genomes, some of them work on ways to change the way genes express themselves. That is going to be really interesting. Then we have two further presentations, essentially reviews of a number of other species under breeding research. In the first, Toby Stovald, a scientist with Forest Genetics Group at SCIEN, will talk about the status of breeding in a number of specialty species. His presentation covers a wide suite of species, including Douglas fir, several non-durable eucalypts, and cypresses. Toby's going to cover past work, where we've come from, current status, and what the future will look like. And maybe he will touch upon genomics and the likes of Douglas fir and eucalyptus. And finally, rounding off this session, we'll hear from the leading redwood grower in New Zealand, Simon Rapley from the New Zealand Redwood Company. Again, this is an update from our industry expert about the status of redwood genetics in New Zealand, and perhaps some commentary also on the opportunity for future breeding. So as in previous sessions, please present your questions as we go. And then at the end of the conclusion, of the session, I invite you to participate in a 15-minute live Q&A session with our presenters. In closing, I hope that you will all go away with a snapshot of how innovative this research is and an idea of some of the opportunities that this is going to make possible for us and our industry. All the experts tell us in the future that the future is one of more risk and uncertainty. So it's comforting to me to see so many of our presenters in this session researching opportunities and ways to sustain and grow our industry in a riskier future. We're indebted to our researchers, and in my view, we haven't yet supported research to the levels that we need to, particularly financially. Enough of that. Now let's hear from our first presenter, Paul Watson.
Good morning, everyone. My name's Paul Watson. Today I'll be providing a high-level summary of the work that we currently have underway at the RPBC and also work underway to look at the RPB strategy going forward. So just by way of a brief introduction, I joined the RPBC back in March. Prior to that, spent 25 years or so working in the pulp and paper industry in technical leadership roles both in New Zealand and in British Columbia. My experience ranges from resource characterization at the fundamental fibre level, including the acquisition of SilverScan 3, through to the mill implementation of state-of-the-art inline wood and fibre quality measurement technologies, and also new product development. This was driven for the most part by working at the epicentre of BC's mountain pine beetle epidemic. And for industry, that mountain pine beetle epidemic is really one of dead trees, lots and lots of dead trees. So the RPBC's mission, just as a reminder, is to breed elite genetic material and provide knowledge, support and tools to continuously improve profitability for Australasian radiata pine forest owners. The RPBC's breeding program has actually been described as a bit of a treasure. It's taken many years and a lot of hard work by very smart and dedicated people to reach this point with radiata pine. And I think the point is very well made. For forest managers, it's fast growing, it's very reliable, and very forgiving over a range of sites. Our current breeding objective is shown here and was developed by committee. The 4 components, growth rate, branching habits, stiffness, and wood density, remain the essence of the current program. Bigger, faster, stronger, and smaller branches, if you like. The breeding program itself is best described as more of a rolling front. So on the left-hand side, annual crossing is followed by seed collection, and then field testing activities, and then scale up. And for a single crossing, this is a 17-year timeline, just to bring new germplasm to the market. Of course, these are happening every year. In terms of our operational program, last year we established 7 field trials and measured 8 existing trials. This year we'll establish a further 5 trials and complete 9 assessments. All trial data is now being uploaded to GeoMaster for easy access to our shareholders. And we're reviewing and improving both tree tagging and our sonic stem measurement technologies, and evaluating the opportunity to implement new technology to replace subjective analysis like branching index. And we're using the latest UAV point cloud technology in collaboration with Indepine and Scion. And we've been collecting tissue samples over the last couple of years. This is for the development of genomic selection with Scion. And this is to address the breeding cycle timeline, which we'll discuss shortly. Recently on the breeding side, we've made major improvements to our data pipeline and genetic evaluation procedures by introducing Bolt software. This software provides a platform for delivering genomic breeding values by utilizing all traits and genomic information. This tool is widely used for animal breeding, and if you're interested in learning more, I suggest you give our breeder Mark Padgett a call. We're also now in a position to move on from GF Plus ratings to the next generation system, which is based on estimated breeding values. And these are widely employed in other fields. EBV information was posted on our website earlier this year, and there will be further communications about our transition plan later on. So our immediate challenges should really come as no surprise. They are, first of all, to deliver improved genetics, which help maximize the productive capacity of our shareholders' real estate, to minimize the timeline between breeding, deployment, and final wood product, to confirm our value proposition. Proof of genetic gain continues to occupy the thoughts of many, to enable greater access to elite germplasm, consider climate change and future breeding activities, and to consider future wood quality requirements and end uses given the 30 to 50 time horizon from breeding to product. And the onus is still on the RPBC to prove that the breeding program is in fact delivering genetic gain. Now, there's a couple of excellent publications out there by John Moore, Heidi Dungy, and Mark Kimberley, which have confirmed the benefits, and then an example is shown here for volume gain of improved versus unimproved material over one rotation. And I'd encourage you to take a look at those publications if you have some time. That said, we certainly recognise that forest owners do need a combination of certainty and courage, perhaps, when choosing to plant elite germplasm. There are long memories out there, but we do believe that the requisite tools and information are now in place. So if we think about it in terms of a call to action, it's really about how the RPBC might better deliver elite germplasm with confidence in field performance and final product quality, and with urgency, while also considering future market and climatic conditions. So at this point, I'd just like to pause briefly and talk about innovation. It's arguably the second most misused word in the English language after sustainability. The definition we used at Paprika and at Canfor was this one, which was coined by the Stanford Research Institute. These guys invented the computer mouse, the LCD, Siri, and NASA Solar Probe, just to name a few. And that is, innovation is the creation and delivery of new customer value in the marketplace that also provides sustainable return to the organization. It's about ambition, courage, and urgency for the RPBC. This is very important because our business spans that implementation space between science and operations, which is generally described as the valley of death. So therefore, an open innovation approach is really a prerequisite for us going forward. The open innovation model is one where a pipeline of internal projects is complemented by carefully selected global research projects outside the organization, while being very conscious of boundaries and, of course, not to become overextended. The program outcomes are commercialised, some of them, some are spun out, and some are just put on ice. So to support our shareholders and to ensure that the RPBC remains a viable long-term business, it's important that we expand our network of program activities, that we collaborate rather than compete, and that we leverage every dollar against other funding sources to maximise the successful implementable outcomes. And we've got to look at every opportunity, not just from within our current cohort or from within our sector. Our core capability will of course remain breeding, but to be honest, business as usual is just simply not an option. And that's simply because there are external factors out there which may be beyond our control, and they do now require consideration and action. Climate change was a significant contributing factor to the endemic mountain pine beetle vector blue stain fungus, which collectively decimated more than 70% of BC's lodgepole pine and remains a significant issue in Canada still today. And as we know, the Europeans and others have their own challenges right now, with steps being taken to mitigate such risks, including the use of alternative species. And then, of course, we all watched with horror last year the devastating fires in New South Wales, and it's a great concern to us. And just for a bit of context, the extent of these fires is shown here on a scale relative to New Zealand. And I'd really like to acknowledge Forestry Corp New South Wales for kindly providing this information. But these two examples both serve to highlight the need to do everything we possibly can to reduce rotation age with no loss of wood quality, and by any means possible. And from a personal perspective, having worked through one dead tree epidemic, that was way more than enough for me. It's also important to bear in mind that we're growing softwood, whereas a learned colleague of mine once quipped we're growing reinforcement. And right now our vehicle is radiata pine. So with the RPBC's board, we've recently reframed our vision statement, which is to deliver prosperity from plantation forests by developing and implementing solutions that mitigate climate change and enable the manufacture of sustainable, innovative products from wood. Our strategic planning exercise is still a work in progress. We've identified our key challenges and our goals and intend to complete the plan for rollout at our December technical meeting. But we do recognise that world-class breeding is not linear. The incorporation of market feedback is absolutely essential. And for example, having spoken with a number of sawmillers lately, this SG10 requirement is currently a significant barrier to entry into the Australian framing market. So what steps can we implement to address this? And in my pulp and paper marketing experience, I'd suggest that these types of challenges will only get tougher for us. Consequently, the breeding objective that we have needs to be a living document. It must consider the future, and it must consider those scenarios or issues which we may encounter along the way. Now, I just want to make note that molecular markers in genomics is a key step linked to all traits in this particular program. And as we'll hear more about in the next presentation, the RPBC, in partnership with Scion and supported by MB, is close to achieving the aspirational goal of reducing the generational interval we discussed earlier from 17 years down to 9 years, and that's through the application of genomic selection. However, I would caution that there is still much work to do to deliver a cost-effective SNP array for the industry. And there's also still work to do to identify SNPs and even genes that are responsible for those highly heritable fundamental traits which underlie the composite outcomes like density and MOE that are key elements of our breeding objective. So our immediate priority is to transition from phenotypic-based selection to one that is genomics-based. We're on track to select seedlings based on genomic breeding values for 2021. And we're also working to develop a cheaper genotyping platform and watching the very promising 21st century tissue culture project with great interest, as that should really help speed deployment. And we'll learn more about that shortly in this session. Our genetic gain priority remains unchanged. And with that genetic gain firmly in mind, we're looking to successes in other species and the like for opportunities that may be applicable to us. And an interesting example obviously here is shown as with ryegrass, these endophytes. And we've partnered with the Tree Lab to identify novel endophytes for pinus growth rate and disease resistance improvements. This project is very complementary to the Forest Growers Trichoderma program. And Brazil's remarkable success with eucalyptus hybrids has certainly proven what's possible through hybridization. Alongside our shareholders Proseed and Matariki, we're developing a pine hybrids program, starting back with the radiata attenuata hybrids, which are currently being deployed. And we anticipate a wide-ranging program in partnership with a number of companies, including Scion. And we also recognise that in the not too distant future, forest certification systems, and in particular FSC, will adjust their stance on genetic transformation. So rather than wait and watch, we need to be ready to apply this technology, or we will almost certainly be left behind, and very, very quickly. And it's going to be interesting to hear more about this later in the session too. But we should not underestimate the size of this particular challenge, quite literally. The radiata genome is an absolute beast, as you can see. It's 8 times the size of our own. To this point, Science teams have done a superb job mapping the genome, and they continue to work hard on refinements so that we will be very well placed to progress the next phase of gene discovery and transformation. Now, I touched on phenotyping a little earlier and the importance of fundamental traits, particularly in their relation to future opportunities. You may not be aware, but there are now 49 plants globally manufacturing variations of these cellulose nanomaterials, which are shown here as nanofibrils. Well, these are deconstructed fibers, if you like. Markets here are growing, and who knows where this is going to lead. And this is just one example of wood products innovations that we do need to start thinking about. And another consideration is that inline technologies to measure fundamental wood and fiber traits are becoming the norm, for example, in the pulp and paper industry. From a scanner for wood chip moisture density and species, to the inline pulp-eye system, which measures all fiber properties, including fiber cell wall thickness and fibril angle, even right down to measuring and counting cellulose nanofibrils. Increasingly, this detailed product quality information is demanded by customers, and not just for those specialty applications either, also tissue, for example. And at this point, I really must acknowledge the work of Indipine and Scion that are undertaking to complete STEM mapping, which will be extremely beneficial for our program. And of course, perhaps finally, DiskBot, which now enables more detailed phenotyping to support genomic selection development based on fundamental traits. So to summarize, pretty exciting and challenging times ahead as we work hard to reduce not only the generation time, but also the rotation age of pine. We will deliver a world-class breeding program by implementing BOLT and operationalizing genomic selection, implementing future-focused phenotyping, and expanding our field trials program. In the short term, we're looking to include more drought-tolerant germplasm from Australia into our program, and seed is actually being imported as we speak. And we want to be able to exchange additional germplasm, so a Pinus import health standard application is being worked on too. Of course, we'd like to reduce the bottleneck to see more elite CP material being deployed. Moving to estimated breeding values should provide more clarity around seedling traits. Hybrids and endophytes offer unrealized opportunities for growth, disease resistance, and potentially in wood quality. And then there's the potential for genetic transformation. So the length of the forest products cycle requires that we keep looking to the future. So we must be very well connected with world-leading science. And most importantly, we must ensure that our shareholders remain very, very well informed. So in closing, I'd like to acknowledge the support of our 16 shareholder companies shown here, and also our research partners, Proseed, Scion, Theta Solutions, University of Canterbury, the Tree Lab, and Indipine. And I'd also like to thank forest growers for the opportunity to address you today. Thank you very much.
Forestry, a major export industry regionally vital for economic development. Forests are sustainable, protecting the environment, the air, the water and the land. Forests, a place to play and a thriving investment for generations to come. Love our forests.
Hello, my name is Gancho Slavov, and I work at Scion, and I will be presenting today about genomic prediction and its applications in tree breeding, conservation, and mitigation of climate change. So as most things in modern science, I would like to first acknowledge the fact that this is the product of teamwork and acknowledge my colleagues, Yaroslav in particular, for running a lot of the analysis, Mark Paget from the radiata pine breeding company for helping conceive many of the research questions, Nathalie and Ahmed for helping clean up the data and making sense of things that were at first quite puzzling, and Heidi for her leadership. This was funded by MB and the radiata pine breeding company as part of a collaboration agreement between Scion and the RPBC. Just very quickly, an introduction to the subject matter of quantitative traits and genetic architectures. This is Pascal's triangle, which for those of you fascinated with probability theory as I am will remember has the binomial expansion coefficients, but that also means that they give the segregation patterns for a trait controlled by a single locus or a single gene. So that's 1 to 2 to 1. For 2 loci, and then 3 loci, etc. And as you see, as a trait is controlled by more and more genes, it starts resembling a normal distribution. And above on the photo is the observed distribution of human heights, which is the genetic trait that has been studied the longest, over a century of history of studying that trait. And again, just a definition that a quantitative trait is a trait controlled by a large number of genes or loci affected by both genetics and environment. And what we mean by genetic architecture is an understanding of the complete set of genes that control a trait and also their interactions. We now know quite a few of the loci that affect human height. This was published about 10 years ago, and back then, the number was in the hundreds. Now it's more in the thousands, possibly even tens of thousands of genes that are known to affect human height. Will we ever know the complete genetic architectures of traits? Probably not. So this is again an example based on human studies that show that to understand, for example, the complete genetic architecture of human height, that may not be possible until the human population has reached 10 billion and we are able to sequence all human beings on the planet. So for practical purposes, we can assume that that will never happen in our lifetimes anyway. But more pragmatic technology has come around that allows us to predict phenotypic traits from a large number of markers across the genome without testing the statistical significance of each marker. And this has been called genomic selection because it allows breeders to make selections early on in the breeding cycle based on DNA data as opposed to testing. How does it work, or how do we know it works? We're currently able to take data sets retrospectively, where we have both phenotypes and genotypes, so DNA genotypes, and then train a model based on the vast majority of the data, based on the phenotypes and genotypes and the statistical relationships between them, and then predict the phenotypes for a subset where we only use the DNA marker data. And then we can calculate the correlation between the predicted and observed phenotypes and quantify predictive ability. That's the, again, we measure that through the correlation of observed and predicted phenotypes. Where is that going to make an impact in tree breeding? Obviously in field trials, where instead of waiting for a number of years before we can collect data on growth, and possibly even more years before we can collect data on wood quality, we can make these predictions very early on, essentially as soon as we are able to get enough needles to extract DNA, which is just a few months. Where are we with radiata pine, and where are others across the world? The implementation of genomic selection, the technology I just explained, is the main goal of the RPBC Scion collaboration agreement. And the radiata pine breeding company has made a very ambitious investment in genotyping. Over 10,000 trees have been genotyped with the current technology, which costs about $30 to $35 per tree. And it has already enabled significant improvements in the pedigree information used by the RPBC. So when we first started looking at the data, roughly 1 out of 4 first-degree relationships was inconsistent between the records and the DNA evidence, meaning that there was likely an error either in the records or in sampling the trees. So by various analyses, we were able to reduce these discrepancies down to under 5%, which is on par with sort of world-leading breeding programs. It's a very, very good indicator, and it has measurable impact on the analysis used to calculate breeding values. We'll see an example of that later on. Assessments of genomic selection are underway elsewhere across the world, but generally on a much smaller scale. So what we would like to know is, can we use genomic selection to make predictions across different breeding populations? Also, can we use it for making predictions within families, particularly within controlled pollinated families, and across entire series of trials, including trials that have not been genotyped? So we have everything we need to answer these questions with sort of convincing confidence because of the large data sets that we have. So just a quick explanation of what we did. We assembled data sets from older clonal tests. So these are 397 and 399 series, a QTL mapping family that we use to test within family prediction, and perhaps most importantly, cloned elites, which is the sort of the focal population for the radiata pine breeding company, but also for seed producers. So in the first instance, what we were trying to answer is, can we train a model based on the older clonal tests primarily and do predictions within the cloned elites? And the answer to that was yes. So just to explain this graph, on the left, on the x-axis, we have the number of genotypes from the cloned elites that were included in the training population, and that varied from 0 to 300, 0 being that we never test anything from the new population. We just make the predictions based on existing populations. And you can see that even with that, we were able to get predictive ability of roughly 0.2 for DBH, diameter at breast height, and just over 0.4 for wood density. But with testing as few as 100 genotypes from the new series, from that new population, those numbers increased roughly by 50%. So now the predictive ability for DBH is closer to 0.3 and closer to 0.6 for wood quality. So again, even with minimal testing, we can achieve reasonably good predictive accuracy. The other graph illustrates how many markers or how many SNPs are necessary to achieve these predictive abilities. And what I think is good news is that we don't seem to need the entire set of 30,000 SNPs that are currently on the genotyping platform. We might be able to achieve that with a much smaller number of markers. So this is just an illustration of random samples. So not taking into consideration the information content of the SNPs, which we could do, just selecting them at random. So even a randomly selected subset of 2,000 markers gives you just about as much predictive ability as the whole set. So similar question was about the ability to predict within families, or in other words, once there is a family identified between promising parents, can we also do ranking of phenotypes within that family? So this is where we trained the model in the cloned elites and older tests, and the predictions within a family that we've generated for QTL mapping purposes. And that also worked fairly well for wood density. You see that again, even with 0 individuals from the family included in the training set, we're able to recover predictive ability of 0.6. Not so well for DBH, where then pursued that, and a separate analysis that I won't present illustrated that in order to do predictions for DBH, one just needs to grow about 20 to 30 individuals for the family. So where that leaves us is that we are able to do within family predictions and genomic selection for wood density without testing any individuals from that family. If we want to also do predictions for growth, we have to test about 20 to 30. And once again, the graph on the right shows that we don't require that many markers to do that. About 2,000 to 3,000 will do the trick. Then another important point is that even with the most ambitious investments in genotyping, we were not going to be able to genotype everything. So a single step evaluation analysis is where we can combine data between genotyped and non-genotyped trials and trial series for predictions. And we tested that, and this is a kind of a complex table, but what it shows is that for the cloned elites, if we just went with the conventional currently available approach, we would get a predictive accuracy of 0.2. If we use genomics and merged genotyped and non-genotyped trial series, we can nearly double that to 0.23. And again, there's a lot of variables in terms of how the genotyped and non-genotyped data is merged, but that's perhaps irrelevant. Great potential there to combine trial series and populations. There's also quite a bit of interest in producing new hybrids, which might be very important for habitats that are not currently suitable for radiata pine. The current timeline for various reasons for developing new hybrids is quite long, something like perhaps 20 to 30 years. But with genomic selection, we think we can reduce that substantially, possibly even half it. And finally, breeding is not, as indicated by my title, breeding is not the only place where genomic prediction will have an impact. We often assume that local is best, including in eco-sourcing, but increasing evidence shows that many trees have actually adapted to climates that are already gone and will continue to change in the future. So what then becomes important is being able to match genotypes not only to the current climates, but also to the expected future climates. And the state-of-the-art approach for doing that is essentially putting a bunch of field trials in different environments and then testing a lot of populations or provenances. This works really well, but it only starts working well when you have a large number of test sites. So genomics potentially allows us to shortcut some of that and make some of these decisions based on smaller numbers of trials, which will be particularly important for indigenous species. So in summary, we know well that we know that genomic selection works well in radiata pine and will allow us to do things in new ways, particularly for wood density, where we can make predictions across populations, across series, and within families without testing any individuals for the family. For DBH, we could do many of the same things, but probably a more conservative recommendation is to test 20% of each new generation in series. But this is still a significant reduction of field trial costs. And a lower cost genotyping platform that hopefully will cost roughly half of what the current one does is under development and will be available next year. And we also have preliminary results to indicate that this technology will work very well in Douglas fir and eucalyptus. So please see Toby Sowell's presentation on that. And finally, a point that the genomic prediction can be used for not only for breeding, but also for mitigating the effects of climate change in both exotic and indigenous tree species by matching provenances to future climates. And with that, I'd like to thank you for your attention and wish you a pleasant conference.
Hello, I'm Russell Burton from Forest Growers Research Limited, and I'm the Program Manager for the 21st Century Tissue Culture Program.
I am Jana Krajniakova. I work for Scion, and I am the science leader for this program, which we are going to now talk about.
Yes, we look forward to taking you on an update of the Tissue Culture Program, which started 1 year ago. This program aims to make production of varietals of radiata pine more cost-effective and accessible to the New Zealand commercial forestry industry. The New Zealand commercial forestry industry has made large investments over very many years developing elite germplasm of radiata pine by means of different breeding strategies, with the aspiration to populate our commercial forests with those trees that create the best value for the industry and continue to grow that value. That is, those trees that enable us to continue to reduce rotation age, give us the best quality of wood within the trees, and maximise yield and productivity. And at the same time, and importantly now, very importantly now, we need to ensure our trees are the most resilient they can be, as with climate change comes a heightened risk of their exposure to pests and pathogens. To fully deliver on growing the value of breeding, we'll need to transform our commercial forestry model from a family forestry model to a varietal forestry model. Varietals are trees with very similar DNA. By focusing on producing varietals of the best trees, we will increase the average qualities of important attributes such as biomass productivity, wood quality, and uniformity. Recent advances in tree biotechnology, including somatic embryogenesis, have enabled the development of a more flexible tree breeding, with genomic selection and deployment strategies over conventional seed orchard approaches. In addition, we expect to get deployment time down from typically 24 years to 6 years, when genomic selection and somatic embryogenesis are combined. This slide shows the process of somatic embryogenesis. The pleasant protocols for somatic embryogenesis of radiata pine start with immature seeds, which are produced by crossing trees that exhibit the traits that we are after. The big advantage of somatic embryogenesis over other methods of vegetative propagation is that the embryogenic cell lines can be kept in a juvenile state by cryopreservation for later use. That means we can field test some of the genotypes and later focus on those genotypes whose performance can be assured, creating a further step change in raising the average properties of the trees in our forest. Our new program seeks to exploit the advantages of advanced somatic embryogenesis approaches and at the same time make a step change in improving the cost effectiveness of the production of varietals. We'll achieve this by using modern bioreactors and robotics rather than petri dishes. There's nothing new about creating varietals via somatic embryogenesis, with varietals produced this way already in commercial application, although in a very small way. Our business case for using varietals in commercial forestry tells us that if we could achieve about 40% of the commercial forest estate, we would realise at least an additional 1.5 billion per annum in value for the industry. What limits varietals increasing their share of new plantings is cost associated with small yields and the high level of manual involvement in the process. Typically, somatic embryogenesis has been based on cultivation on semi-solid media, with several transfers required, as the media needs to be adjusted based on the stage of development of the somatic embryogenesis. These barriers can be overcome by using bioreactors instead of petri dishes for growing the embryos, coupling this with automation and robotics, and as a consequence we can build a very high throughput propagation factory. This is the basis of this new tissue culture program, which is now 1 year old. Essentially, our plan is to move from the petri dish-based propagation to high throughput bioreactors and couple this with automated embryo sorting and germination. What our program will do will be to replace many of the traditional operations by using bioreactors, specifically a bioreactor called a temporary immersion bioreactor. They called this as the liquid which provides the growing stimulants, temporarily floods, then empties from the reactors. The environment inside this reactor can be very closely controlled and changed as required to induce the embryos to develop and produce hundreds, if not thousands, of plantlets. This creates better access to the key components in the nutritional media, higher proliferation rates and increased production of the somatic embryos. There are a lot less manual steps and provides scalability through the exploitation of robotics and automation. This approach offers several advantages over manual handling and using petri dishes. The use of liquid media also has an advantage in simplifying the manual handling. When the somatic embryos are ready, they can simply be transferred into another vessel, where the wonders of artificial intelligence and robotics can decide which are the good quality plantlets, remove those which are not properly developed, and then transfer the good plantlets to a soil plug, ready for the next stage of their journey to the nursery. Although we are pioneering this for radiata pine, aspects of the approach we have just described is already well advanced for some other species. We have forged some relationships with other organisations internationally, and as a consequence been able to leverage off their knowledge as we build a system that works for radiata pine. This last year, the program's first year, has seen us consolidate and formalise those relationships. And in spite of the challenges thrown at us by COVID-19, we have succeeded in building at Scion the first ever facility purpose-built to house the temporary immersion bioreactors focused on the tissue culture of radiata pine. Let's see it in operation.
I would like to take you now for the virtual tour to Scion laboratory dedicated to bioreactors. This laboratory has two independent rooms, bioreactor room and preparation room, where we have laminar flow for work in sterile conditions. Now we will be entering to the bioreactor rooms where we have control airflow. Then temporary immersion bioreactor setup systems work with compressed air, which is going to be explained later on. The bioreactors are connected with automated control with computer software called SoftRun. Now I will talk a little bit about technical design. Scion has compressed air running below the floor, and we have moved it up and compressed air is now in a steel pipe carrying to the air filter, and then we control also the pressure. In present moment, we work with the pressure 7 kilopascals. Then filtered air enters the solenoid valves, which could be digitally or manually controlled. Software opens and closes the valve according to user's instructions. When valve opens, filtered air enters the right tube. And this supplies a whole bank of bioreactors. The bioreactors are controlled also manually using manifold. Therefore, we can switch on or off bioreactors which are not in present use. Tubing are connected to bottles containing culture media. Compressed air passes another air filter unit. And then pressure of compressed air displaces media volume. Media flows upwards through another filter and it is reaching bioreactor. When the solenoid valve closes, media drains back to the bottle. Now we are going to demonstrate how we work with plant material. For education purposes, we have used blue dye. When material is in bioreactors, then also media is poured into the bottle. Everything has to be closed and sealed properly, then moved to bioreactor's shelf, and compressed air tubing is attached with bioreactor. The connection between bioreactors and the computer is done using special software, SoftRun. Here we demonstrate how we operate using Bank 2. We are going to set our software for 2 immersions, which are going to start at 9 in the morning and then at 9 in the evening. And the duration of immersion is going to be 25 seconds. It is time when the solenoid valves will be open. We are now demonstrating what is happening when the solenoid valve is open. That medium goes up, it is reaching the platform which is holding plant material, and it is bringing fresh medium. Plant tissue is nourished with the new medium for a period of 25 seconds. It is really important to control that the medium will not rinse the plant tissue into the medium. When the immersion cycle is over, as you can see, medium drains down into the bottle. And this cycle is about to be completed. Now, short demonstration how embryogenic material look like after 14 days being cultivated in bioreactors. And these are our current experiments.
As we indicated, we have a large number of partners and collaborators involved in the program. We're able to leverage off a lot of that knowledge by working with organisations such as the Georgia Institute of Technology, the Finnish Natural Resources Institute, and so many other organisations. Most especially also the New Zealand industry, which are providing a lot of input into the program design and development and bringing their experience within as well. Also acknowledge the support from the Forest Growers Levy Trust and the Ministry of Business, Innovation and Employment. So in conclusion, our program aims to add some significant new tools and knowledge to our industry by harnessing the power of bioreactors, automation and robotics in the production of varietals using somatic embryogenesis, making this accessible to all those in the industry by improving cost effectiveness and reducing deployment time. Another key step is building our knowledge with industry and adding value across the entire industry. We thank you for your time and we hope you found this presentation of interest. On behalf of Jana and her very extensive science team and our industry support team, I thank you for your time and interest.
Sustainable planted forests. They're a natural tool to soak up atmospheric carbon in trunk and building. Forests protecting our fragile landscapes and cleaning waterways. A habitat of choice for many of our endangered wildlife. Love our forests.
Hello, welcome to my talk. My name is Clement Saltana from the School of Forestry at the University of Canterbury, and I'm working on durable eucalypts together with the New Zealand Dryland Forest Initiative. The title of my talk is Breeding Durable Eucalypts for Wood Quality, and it's really an introduction in the NZDFI breeding program. So it deals with the breeding program, which is at the center of the NZDFI, and we can ask the question, why is the NZDFI structured around the breeding program? It's because we think we can make large gains by breeding. And it's very obvious if we look at agriculture as an example. On the right, you see a banana and the wild banana on the right, and the banana which we now commonly get in the shops. And you see huge advances being made, and there's no reason to think that trees are any different to that. It's just that the breeding programs are in earlier stages in forestry or with trees, and very often there's wood properties not even included. But I would argue that it's very important to include wood properties into breeding programs, because if you go back to the agricultural example, that we can't sell any bananas if they do not taste good. Likewise, we can't get a lot of money out of trees if the wood is not up to its specifications, and we need to sell it for firewood or some lower value products. So we need to think about wood quality, and wood quality is really a desired set of wood properties for a certain application. It has no meaning if we do not put it into context to a product. So we need to know, what does the NZDFI actually want to grow wood for? What are the target products? And we find that information in the NZDFI strategy. And there's just a little cutout of the NZDFI strategy, which states right down here to produce naturally durable strong timber for laminated veneer lumber, LVL, decking, and posts. So these are the products which we in the NZDFI try to target. So now we need to think, what are the properties which are essential for that? And this is for posts, it's ground durability, so that they last a long time in the ground and not decay. It's color for sawn timber, and for LVL, that's mechanical properties, size, stiffness, and strength. Additionally to the product properties, there are also some issues which we often have when we process timber. And for the eucalypts, we know that there are growth stresses in the trees which impact solid wood processing, and we know that it's sometimes very difficult to dry the timbers without getting collapse defects during the drying of eucalypts. So we know what we are looking for. Now let's see what species which we can use and what species are in the NZDFI breeding program. And it's largely based on 3 species. That's Eucalyptus bosestuana, Eucalyptus quadrangulata, and Eucalyptus globoidea. And you can see they are quite durable species. The hardwood is of high durability, class 1 or class 2, lasting more than 25 years in the ground. And they're also exceptionally stiff, so good for LVL or structural products, and 20 gigapascal is probably close to the theoretical maximum you can get for wood. They also have color, and you see there are the 2 minor species we have in the program. We have them in for the color, also because of the durability, and we think that we might be able to create hybrids between the species and therefore increase the genetic diversity in there, and also the properties in the wood. So here have we species, we have the products, we are knowing the traits, where to look for, and they are the typical forestry traits like growth and form and health and propagation. I'm not talking much about these here. Form, we can almost think about a wood quality because it determines the yields of our timber. But then there are growth strain, very important, and the hardwood properties, because only the hardwood has color and only the hardwood is naturally durable. And really, instead of normal growth, we want to know how much hardwood is in the trees. So the ranking of these individual traits will be different priority for the different species. Some have naturally better form than others. Others are naturally more durable than others. So if we include these traits into a breeding program, we also need to have a means to measure them. And measuring growth strains is notoriously difficult. With the common technologies to measure growth strains, it takes you minutes or hours to measure a tree. And we can't really afford that in the breeding program, because the breeding program really lives off large sample numbers to as the phenotype many trees. And for growth stressors, what are the problems? So if we do solid wood processing, we cut the tree, we release the stressors, and we get these huge deformations if the growth stresses are high. Like here, horizontal bandsaw, splitting a log, and it has completely deformed. Or if we do rotary peeling veneers for LVL, you get these end cracking due to the growth stresses. Now, the School of Forestry came up with a method to measure trees quickly for growth stresses, and it's less really measuring the growth stresses, but measuring the problem, the deformation we get caused by them, and we call it the splitting test. We grow trees only 1-2 years to a certain size, a couple of centimeters thick, and then we split them all along the stem with a bandsaw, and we measure the opening. If there's a large opening, it's a bad tree. If there's a small opening, it's a good tree. And we measure the problem directly, and we can do that very quickly on a tree, and therefore cope with large sample numbers. So in a SFF, Sustainable Farming Fund project, which has now been successfully completed, we screened almost complete breeding population of the NZDFI. What wasn't screened was Eucalyptus globoidea, but you've seen we've planted close to 20,000 trees, which we have assessed for growth strain, and we have the data, and we got data like that where we can rank the individual families that seed from one mother tree according to growth stresses here, and here it's quadrangulata, and you see the bad families up here have a growth strain of 2,000 microstrains, while the good trees have 25% less growth strain. And if you plant those trees, we will have an improved resource with less processing problems, with higher yields. Now for hardwood, we need to do different tests, but the really what we want in our trees is the hardwood. We are not really making much money out of the sapwood, because only the hardwood is durable, and the hardwood varies a lot between trees. So here you see Eucalyptus bozestuana trees, all the same age. The hardwood is highlighted red, and the largest trees, which we would have selected if we just select for diameter, doesn't have the most hardwood, almost nothing. While smaller trees can have much more hardwood, and that's really where the value is. So we need to measure hardwood quantity as a growth trait, and we also need to measure how good the hardwood is. And again, we see a huge variation. We see some trees have a lot of extractives in the wood, others have very little extractives in the hardwood, and the ones with a lot of extractives will be the more durable timber. While if there's no extractives in the hardwood, nothing gives the color in there, then it's very unlikely to be durable. And now we need also means to measure it, like for growth strain. These traits are not straightforward to assess. We need some wood samples. So the first thing we did, we developed a corer, and we came up with Color Innovation came up for us with this corer. It's battery powered, it's lightweight, it's fast. We can get a decent wood sample non-destructively out of our trees within a couple of seconds, and then we can look at them and assess how much hardwood is in the trees. So that's straightforward. For the hardwood quality, how durable it is, it's more difficult, but we also came up with a way how can we do that quickly. We can do it with near-infrared spectroscopy, NIR, where we're basically looking at the color of the wood, but the color in a wave numbers and in wavelength which the human eye can't see. And if we do that, to take the NIR spectra, that takes only a couple of seconds, and we have calibrated our machine for the extractive content measured conventionally. We get really good correlations, and we find trees with more than 16 up to 20% of extractive of the masses is wood. They are durable, and the other ones with low extractive content are very unlikely to be durable. And we can predict them and find those trees quite accurately. So we have ways to assess large sample numbers, and we need to be able to assess large sample numbers because the NZDFI breeding program is reasonably large. So if you look at all the different trials we have put in of different species, at the moment there are 70,000 trees out there, which hardly have been assessed. Some will be assessed. There are many more trees planted because the trees have been thinned and are going to be thinned. And you see they are distributed all over, or predominantly in the east coast of New Zealand in the warmer regions. Okay, how far are we with our phenotyping efforts for Bosestuana? We have a large part of the breeding populations assessed for hardwood traits and also for growth strain. The last trials are to be worked on this year and the next year. Quadrangulata, we have assessed for growth strain. We are starting to do hardwood assessments this year, and for global year, we have started for hardwood assessments. We will finish these hardwood assessments as well this year, at least that's the plan, but we haven't assessed them for growth strain. From the data we already have, we have been able to make selections, identify superior genetics, which also includes growth and form data, not only the wood quality traits, but so wood quality and forestry traits. So these selections have been made, and those trees are now also propagated under 1 billion tree funded program, and that has just started. And by 2021, next year, we will have produced 300,000 elite plants, so with improved genetics of Eucalyptus bosestuana and Eucalyptus globalia. That's a mixture of seedlings and clones, and they will be available under the Xylogene brand, so that the forest grower can be sure that they have the best planting material available when they establish those plantations. Okay, what is the future for the NZDFI breeding program? We need to continue the phenotyping. We need to go to the next breeding cycle, make it a second generation, put in those trials. We want to continue our clonal breeding program. We're looking at the genetics, economics, right? There's a lot to be done, and we have secure funding till the end of 2022, and we are now in the process of trying to secure continuing funding for this ongoing work, not only in the breeding space, but also in the related areas of processing the wood, of growing the wood, of tree health, and so on. So this work wouldn't have been possible with a lot of collaborator, PhD students and master students at the School of Forestry, a large team at the NZDFI, and funding from the government and the industry. And I would like to thank you all for watching, and I hope to see you soon in person.
So good morning, my name is Glenn Fulby and I'm a research leader at Scion in the area of plant biotechnology. And today I'm going to tell you a little bit about several technologies or biotech-based technologies which we believe that over the next few years will add real value to conifer breeding programs. And now to start the presentation. So today, as I just said, I'm going to talk about the transformational opportunities that several new breeding technologies offer to conifer breeding programs. During the presentation, I will give some examples of how the technologies described are being used in the MB Winning Against Wildings program. For those of you who are not familiar with this, it is a multidisciplinary project looking at all aspects of the wilding problem in New Zealand, from ecology through to providing solutions to existing and future wildings. Some of the work in this programme, particularly work focusing on producing low coning or sterile Douglas fir, is supported by the forest growers. The key aim of the project is shown here, which is to use biotechnology to stop essential genes or processes that are required for coning, and thus prevent new wildings. This will enable future plantings of Douglas fir, which, although it is prone to wildings, is highly productive in parts of the South Island. I'm going to describe 4 new breeding technologies that are either starting to be used or already in use in agricultural plant breeding programs, and also provide exciting opportunities for forestry. The first 2 technologies, transgenic modification and gene editing, alter the DNA sequence. In the case of transgenics, this has led to organisms produced using it being classified as GMOs. Whilst gene editing, because of the nature of the changes made, is classed in much of the world as a non-GMO. Currently, this is not the case in New Zealand, but I think over the fullness of time, New Zealand will align with global regulations. The other 2 technologies, exogenous RNAi and epigenetic modification, do not alter the DNA sequence itself, but they alter the way that genes work, and because of this, are globally considered to be non-GMO. Transgenic technology is probably what comes to mind when you think of genetically modified organisms or GMOs. It involves adding new genes or DNA, likely from a different species, to make transformational changes in phenotype. It has had a huge uptake in agriculture, where the majority of crops such as soya and cotton are now GMOs. It can be used widely across different traits, with pest resistance and herbicide tolerance the most utilised, but it is increasingly used for consumer quality traits as well. It's an area where Scion has done a lot of work over the last few years, particularly looking at sterility, growth and wood processability. It's one of the key technologies we have used in the Winning Against Wildings programme, and shown here are some transgenic Douglas fir trees growing in Scion's GMO glasshouse with modifications designed to produce sterile trees. Next, I would like to say a little bit about gene editing. Gene editing allows you to change a gene in a highly specific and targeted way to create a designer mutant with an advantageous phenotype. You don't have to add new DNA, and the changes made are of the same type as occur through chemical mutagenesis, which is a non-regulated process, or indeed through evolution. As I mentioned earlier, this has led to much of the world not regulating this technology as a GMO. So how can we apply gene editing in forestry? The cartoon you can see here outlines some of the examples being used in agriculture. You can see that the applications are very similar to those for transgenic technology, with a focus on stress tolerance and disease resistance. In forestry, yield improvements would come from growth and wood quality traits rather than grain traits as shown here. An important observation is that the first gene-edited crops have now been commercialised, and these are not being regulated as GMOs. From a forestry perspective, gene editing is a really exciting technology with huge potential. For this reason, Scion has put a lot of effort into developing it in conifers over the last couple of years. I'm pleased to say that we've had success with this, although there's still quite a lot of work to do. In this picture, you can see some gene-edited radiata trees growing in Scion's GMO containment glasshouse. These have had a cell wall gene edited, and we've looked at the cell wall characteristics and found that they are different. We also have some cell cultures of both Douglas fir and radiata with edits in genes we think are essential for reproduction, and so should produce sterile trees. Over the next few months, these will produce seedlings that will be transferred into the glasshouse. The next two technologies I will describe are more recent, and you're probably less familiar with them. These alter the way genes work, but they don't alter the gene's DNA sequence. Because of this, they are not considered to be GMOs. The first of these, exogenous RNAi, uses a cell's own molecular machinery to inactivate genes. The natural role of this process includes things like detecting and destroying non-host RNA, such as from viruses, and regulating gene expression. The diagram shows one of the ways in which this works. In simple terms, a gene, which is encoded by DNA, is copied to produce a messenger molecule called messenger RNA. This messenger RNA is then used as a template to produce a protein, and proteins generally carry out functions within cells, such as enzymes. One of the ways that cells regulate this process is by producing regulating RNAi molecules, here RNAi. These molecules interfere with the messenger RNA to degrade it and ultimately stop the protein being produced and turn off the gene function. There is a lot of knowledge about how this process works, so we can design artificial RNAi molecules to turn off genes. This can be very targeted, as we can design RNAis against individual genes whilst not affecting others. This technology has been widely used as a transgenic tool, but we now know that you can treat a plant with RNAi which has been produced outside the cell. For instance, spray it with a solution. The RNAi then enters the plant and is able to change gene expression. Because this effect only lasts whilst RNAi is present, it's a transient process. The biggest challenge with using this technology is the ability to apply RNAi at the right time and in the right amounts. So far, the main application of exogenous RNAis has been in the development of RNAi-based pesticides, where the target genes are essential genes from insects, fungal or viral pathogens rather than plant ones. These RNAi molecules can be applied to a crop, and attacking pathogens take up the RNAi. This then turns off the target gene, killing or disabling the pest. As you target a particular gene, this is a very specific process and does not affect non-target organisms or indeed the host plant. RNAi also has very low intrinsic toxicity. The RNAis can be formulated with carriers, such as nanoclay, to give slow release and prolong pesticide activity. RNAi can also be used to influence plant gene expression, and I will say a little bit about one possible use for this later in enhancing tissue culture performance. We're also interested to see if we can use the transient nature of exogenous RNAi to turn off genes that repress cone development in Douglas fir to allow precocious coning. If we can do this, we can use it to test the effectiveness of the sterility constructs that we've made in the Winning Against Wildings program. The final technology that I want to talk about today that can potentially alter phenotypes without changing the DNA sequence utilizes epigenetic modifications. I won't go into detail about this other than what you can see in this diagram. You're most likely familiar with the concept of DNA being a double helix, as shown here. But in the cell, the DNA has a much more complex structure than this. The DNA is further packaged around proteins and condensed to form chromosomes. And the DNA in its packaging can have various chemical modifications. And these modifications affect whether and how well genes work, for instance, turning them on and off in response to environmental challenges. A major difference between epigenetic approaches and the RNAi approach we've just talked about is that epigenetic changes can be very long-lasting and can indeed be heritable, passing to the next generation. But unlike DNA sequence changes, they are dynamic, so can be altered in response to environmental factors. That's all very well, but how can we utilise epigenetics in breeding programmes? One way is that as we better understand epigenetic changes, we can use them to predict outcomes and use this knowledge as a tool to select desirable phenotypes. There's actually a lot of work around this in human health, as epigenetic changes that occurred earlier in your life, or indeed in your parents or grandparents, alter your risks to various diseases later in life. We're also starting to understand how we can modify epigenetic profiles to change the way genes work. Here you can see a simple example of how this might work. In nature, epigenetic changes can be primed so that a stimulus can prepare the plant to deal with a stress later on. This makes them better able to respond to a stress. We can achieve the same effects using priming stress, chemicals, or molecular tools to give long-lasting phenotypes to things such as drought tolerance without altering the DNA sequence. For my final couple of minutes, I would like to give an example of how we might use these two non-GMO technologies using improvements in tissue culture to outline this. As you might have heard earlier, tissue culture is an enabling technology for the mass propagation of trees to support clonal forestry. It's also the route we use to propagate biotech trees. One of the key steps in tissue culture is the production of embryos from cell cultures, which are then germinated to produce trees. This is the step you can see on this slide, with an example of it working well. Unfortunately, the results of this process are very variable. Some genotypes just don't like doing it, meaning that a lot of lines need to be screened to identify good genotypes, and these may not be the best performers in the field. Another issue is that as the cultures get older, they lose the ability to produce embryos. So what you often see, unfortunately, is more like this. We know that both epigenetic changes and gene expression play important roles in how genotypes perform in tissue culture. This opens up the opportunity for screening to identify and select genotypes that are likely to be performing well in tissue culture, reducing the need to do time-consuming testing. But we can also start to think about using exogenous RNAi and modifying the epigenetic profiles to make increased numbers of genotypes more amenable to tissue culture and clonal propagation, thus widening the group of genotypes where we can carry out this process. Exogenous RNAi is particularly suited to this because it has the transient effect we described earlier. So you can add it to cultures when it's required and encourage them to perform better. And when you've done this, you can stop the treatment and the trees will then grow with no effects from the RNAi you added. And just to finish, I hope that I've been able to show you that several existing and emerging technologies are going to provide some really great tools that will enable transformational changes in forest breeding programs. Finally, thanks for listening today, and I'm happy to answer any questions you might have, either in the discussion session that's going to follow, or indeed through my contact details on the final slide.
I'm Toby Stovald from Scion. I'm presenting today on the status of specialty purpose breeding in the SWP program. In the Specialty Wood Products Research Partnership, we focus on three main areas: Douglas fir, eucalypts, nitens, regnans and fastigata. The cypresses, we do loose tannica, macrocarpa and hybrids. The SWP program also does durable eucalypt species, but that will be covered in a different presentation. Moving on to Douglas fir. Douglas fir breeding started in 1957 and 1959, mainly with provenance material from the Washington, Oregon areas. There were more trials in the 1970s, 1990s and in the 2010 era, including a major introduction of Californian provenances in the mid-1990s. Program has also established large plot genetic gain trials in 2012 to validate the gains from various seed orchards and generations of material. In 2018-2019, new trials were established which incorporated material previously being run in parallel by Earn's Law 1. More recently, genomic research has started using two trials. This involved collecting DNA from 2,200 trees, which were then genotyped on an axiom array, and this was in collaboration with Glenn Howell from Oregon State University. And we have 2,000 more samples awaiting analysis. So if you look on the right, the two trials were Kaingaroa and Gowan Hills. If you look at the chart in the slide there, the dark towers are the prediction accuracy of the breeding values when we just have the phenotype data, that's data from the tree, and the grey towers are when we add in the information that comes from the marker analysis. So in all cases, diameter, needle retention, stiffness, we got a better result by adding in marker data to the pedigree data. This year we'll use a very similar approach to estimate Swiss needlecast breeding values for most of the grafted parents that are in the various orchards around the place. Moving on to cypresses, I'll cover loose tannica, macrocarpa and the hybrid work that's happening. For loose tannica, breeding trials began in 1984. We had some failed second generation trials in 1998, which was a combination of goats and a not very fertile site. The second generation was repeated in 2006, and then recently in 2017, a third generation of tests was established. Along the way, a clonal program was initiated in 2002, as cypresses are amenable to propagation by cuttings. Currently, the clonal program is parked. We're not continuing on with that. From all generations of this research, the best material has been grafted to orchards, so good quality seed is available for those who wish to grow loose tannica. Macrocarpa, the first breeding trials were 1984 and a second generation in 1999. Similar to loose tannica, we started a clonal program in 2002, and also this program is currently parked. We're not investing in creating more clones. For this species, interest declined due to canker issues and planting dropped away. However, we noticed some tolerant trees in our trials. So in 2010 and 2011, we selected some parents that looked like they were tolerant in trials in Guavas, Longmire and Birch Hills, and these were grafted to the orchard of Proseed. Over time, the seed orchard flowered and developed seed, open pollinated, and pro seed, did some control pollinated seed. And in 2018, there was enough interest in SWP to get this material out. So we established 3 new progeny trials in 2019, Tarawera, Manawatu and Foxton. And we'll be monitoring these to see if they get canker infection, looking for tolerant families. Finding tolerant families will rebuild confidence in macrocarpa and get people to plant it again. Cypress hybrids, the breeding plan we have calls for hybrid swarms, which is everything crossed with everything. Our main interest is in hybrids with Nucatensis, which would add durability. Nucatensis is class 2. Pollen was supplied from the BC breeding program in collaboration with John Russell. We also used Arizonica and Guadalupensis, thought to be tolerant of canker as well. And we crossed those to our best Lustanica and macrocarpa from our progeny trials and clonal programs, and also with them every combination we could get. Evaluation trials of this material were planted in 2009 and then again in 14, 15, and most recently in 2017. So far, we've evaluated 130 hybrids. 30 of those we identified early as being good in the nursery, they're good propagators. And from within those 30, we released 6 new Avenzii type and 6 new Leyland type to the market so that nurseries could grow store beds and have this material available for growers. Most recently in 2019, we added 2 sites of these new release material to look at matching species or clones to site. And that brings to a total of 8 sites we have with these 12 released hybrids. We have 150 additional hybrids yet to be evaluated. And we recently got some input from the Farm Forest Association through their Cypress Development Group on what they thought would be ideal types for growing. Cypresses, what does the future look like? We need to identify canker-tolerant macrocarpa that will rebuild confidence in growing the species. We'll maintain the Lustanica breeding program, which is pushing on with growth form and monitoring canker. And most importantly is learning about the heartwood and durability of these new hybrids. The hybrids have a lot of potential, and testing to see how much durability we may have gained by bringing in Nucatensis. We need to investigate that and quantify it in the future. Moving on to the eucalypts, I'm calling them industrial eucalypts because they're not durable, and certainly our initial interest was for the fibre side of things. There will be some discussion about solid wood products from these species in another talk, and our species are Regnans, Fastigata and Nitens. So Regnans breeding started in 1978. The current breeding population size is around 100 families. The last round of tests we put in were 2009 and 2010. The population is currently sitting in 3rd generation. We've assessed these trials for growth and form, and following on from that, new material was grafted to orchards. As a backup to increase possible seed supply, if there was more interest in Regnans, we've established some gene resource plantations, and we plan to convert one of the trials into a seed stand, just in case the seed orchards can't meet existing demand. Fastigata, very similar story, started in 1979. This population's a bit bigger, it's 200 families. So we've used a rolling front strategy, which we split the population into 2 groups, which helps spread the resource requirements for testing. So the first half, which was 2 trials, has been assessed for growth, form and stiffness. And just recently we completed the second half, which was another 2 trials, and we'll be reporting on that later in the year. And as a result of that report, we expect to identify some new orchard candidates. Seed orchard seed, yep, it's available from the previous generation, and similar to Regnans, we will convert one site to a seed stand as backup in case there's more demand for the seed. Eucalyptus nitens, this is our one we spend most of our work on. The previous breeding population turned over in 2014. It's a rolling front strategy similar to Fastigata. We have about 220 families involved. We've done quite a lot of wood properties studies in one half of this population. We felled 780 trees. We looked at growth strain and shrinkage. And from each tree that was felled, we collected DNA for some genomic studies. We identified a bunch of new parents for seed orchard use, and further work will be to use the SION spot to create NIR data and create some models for non-destructive sampling. For the genomics, 700 trees, again, SNP chip was used, and similar to Douglas fir, the accuracy of the predictions was improved. If you look in the chart, the black towers, again, are just the pedigree or the phenotypes, and the grey towers are phenotype and the marker information together. So we had improvements in diameter, growth stress, shrinkage, and density. Another thing with nitens is it's limited by Propsis. So with the cooperation of Southwood Export and SION collected adult beetles from Poronui and Taupo. They were segregated into pairs and released into the forest. November 18, we had to wait, checking monthly that there had been damage worth assessing. And in May 2009, we did. We used a score from 1 to 10. 1 was no chewing, 10 was heaps. Turns out it's heritable at 0.15, very similar to diameter. There were some differences between orchards and other sources used, which means we've got the potential to select for browsing tolerance. The trial won't be sprayed this summer, so hopefully we can reassess it. If you look at the graph in the right-hand bottom corner, there are parents that are above average for growth and less browsed. Why does FGR and SION invest in these? Well, 90% of the forest is radiata, so it's risk mitigation. Some sites are not suited to Douglas fir, the cooler sites, and some species are better for the chip market, and that's our industrial eucalypts. Specialty timbers, cypresses, that's a good alternative for radiata pine, and availability. There's good quality genetic seed from the program through the various orchards at ProSeed, Earnslaw One and Southwood Exports. And thank you very much.
Our planted forest industry, challenging to be our second primary export earner. Woods, environmentally good in plant and product for building and landscapes. We're great in technology and science, selecting stronger, straighter trees, safer ways to harvest them, cleaner and earthquake-resistant ways to build. Forests, regionally vital for economic development, creating all sorts of jobs. A prime future investment for our nation. Love our forests.
My name's Simon Rapley. I'm going to give an update on redwood genetics in New Zealand. There are now more than 10,000 hectares of redwood forest in New Zealand, and we know from tree sales from the nurseries that there are more and more trees going into the ground each year. People are interested in redwood because it's a high value timber. Redwood has other traits such as the ability to control erosion better than almost any other tree on steep hill country. And redwood is very well suited to uneven aged management. We also have government policy now encouraging greater diversity in the forestry sector, and redwood is recognised as a viable commercial forestry crop in New Zealand. I am a registered forester with New Zealand Institute of Forestry. I have been growing redwood for a living since 2006, and as a hobbyist, just looking at the photograph there, I started working with redwood back in 1999 on some family land. My current role is the Managing Director of the New Zealand Redwood Company. Interesting for us to look at the situation in California, which is the natural range of redwood. Foresters there have been considering redwood silviculture for a lot longer than what we have here, and have come up with clear strategies. And also for New Zealand foresters and researchers, there are some important differences with redwood silviculture than most of what we do here in New Zealand. And firstly, redwood cutovers don't require replanting. Redwood stumps coppice sometime after harvest. There's a small percentage that don't, but most of them do. And the next crop grows very rapidly off an established root system. And the photograph on the right shows a cluster of stump sprouts that would be 50 to 60 years old, or 50 to 60 years following harvest. Inside that little ring of sprouts will be an old rotting stump from the original old growth tree. And the commercial cut in California is based on these regenerating sprouts now. There's virtually no old growth harvest anymore. The Californian foresters do plant redwood, of course. Redwood occurs in mixes with various other trees, and following harvest, they like to put all of the land back into redwood on account of its high value and ease of management. Traditionally, they've used seedlings for this interplanting, but for a number of reasons, including unreliable cone production and low germination of the seed, there's been a shift to clonal propagation. The last good seed crop was 2009, following wildfires, and the foresters are hoping that there'll be some good seed production next year following the fires that they have in California at the moment. Redwood flowers in the winter, and although California may get us a similar rainfall to New Zealand, none of it falls in the summer, it all falls in the winter. So their winters are cold and wet, and just not good conditions for pollination of the flowers. Another reason that's driven the shift to clonal propagation is that redwood is one of the few conifers that can, where mature trees can be cloned. And of course, that gives the opportunity for rapid deployment of improved genetics. Breeding programs aren't necessary where desirable parents are crossed and seed with its inherent variability is produced. Mature trees with the desirable traits are identified in the forest, and they are cloned, and tissue culture labs can literally produce hundreds of plants in a relatively short period of time. The California growers have recently formed a tree improvement co-op. There are 20 members, and that is pretty much all of the redwood growers in California. They have engaged the services of Dr. Keith J. Wickrama, and if that name seems familiar to you, maybe it is. Keith was with the NZFRI for a while. The co-op has almost 200 genotypes under evaluation, and all of its members are actively bringing material into it for evaluation. It is worth mentioning at this point that they do have different selection criteria to New Zealand. To a Californian redwood forester, all redwood is good redwood in terms of timber quality, and they have no interest in selecting for wood properties. The New Zealand situation is that we have 6 suppliers of redwood planting stock. 4 of them are forestry nurseries, and they are some of the mainstream nurseries. We also have 2 forest management companies who both have an interest in genetics and access to improved genetics, and they're contracting nurseries to grow redwood planting stock for their own planting programs and for sale. These 6 suppliers have production in the range of 10,000 to 250,000 trees each year. All of the nurseries are using seed to some extent, and after contacting these nurseries recently, I was pleased to find out that none are using New Zealand-based seed anymore. There are well-described problems with New Zealand-derived seed, and probably the best example is seed from the Memorial Grove in Rotorua, which is a stand of magnificent redwood, but we believe that most of the trees are brothers and sisters or closely related. Possibly the original seed came from a single tree that was felled for cone collection. But anyway, although those trees are good examples of redwood, their offspring have problems with inbreeding and are less vigorous, and there's a belief that there are compromises to wood properties as well. The largest supplier of redwood declinal stock is producing containerized trees derived from tissue culture. The little flask in the photograph is redwood tissue culture trees as they're being transferred from a lab where they're multiplied up to the nursery, which grows them on to a plantable size. The selection criteria are growth and form. I mentioned before that the Californian foresters are not interested in wood properties, but the New Zealand growers think that with the ability to clone redwoods and bring them into production rapidly, that all traits of interest should be considered in the selection process. And the second trait of interest to the New Zealand foresters, which is of no interest to those in California, is epicormic sprout growth. We prune out redwoods in New Zealand. Virtually everyone who's planted redwood is intending to prune them, whereas in California they don't. They have very little in the way of management inputs, and so they don't see any problem with epicormic sprout production. Now, the largest supplier of tree redwood planting stock in New Zealand is actually a member of the California Redwood Tree Improvement Program and has access to genetics from that program. And because of the interest in wood properties and epicormic sprout production in New Zealand, the elite trees from the Tree Improvement Cooperative are evaluated in New Zealand for those. The foresters in California, as a recognition to what we're doing in New Zealand, collect wood samples from the parent trees, which are shipped down to New Zealand for testing. And unfortunately, with epicormic sprout production, we have to wait till the trees are pruning age in New Zealand before we can evaluate them for epicormic sprouting. There is really has been no attempt at redwood breeding as we think of breeding. The focus is on clonal forestry, which has some important differences. We simply go into the forest, pick out trees that are bigger than their peers. We can take increment cores and have the heartwood tested for natural durability, basic density, any of those things. We have one supplier of redwood planting stock who has established seed orchards in New Zealand, but they are not yet in production. And we have ProSeed have been working with redwood and attempting unsuccessfully, unfortunately at this stage, controlled pollination. So there is that is a good strategic approach for tree improvement in New Zealand, is crossing parents with desirable traits and then creating a New Zealand landrace from those. But so far that's fairly early stages, and again the reliance is on clonal production. And this is the final slide, and it just illustrates the issue of epicormic sprout production. The tree on the left is just a random seedling that was pruned as part of a trial a number of years ago. It does still have a few branches on it, but you'll see that there are a whole lot of small branches and epicormic sprouts that have to be removed if a forester wants to produce clearwood in that butt log. And with redwood, they will grow back. Following pruning, some trees that we describe as hairy will, even if you clean them up like the stem on the right, the next time you see that tree, it will have sprouts up and down the stem. Now, the cost of pruning that tree could be as much as twice the tree on the right. And if, when the pruners come back in to do the second lift, they have to take off a whole lot of sprouts again before they do the second lift, then you can see that it wouldn't take much to double the cost of pruning operations. So we are very interested in this trait in New Zealand, and as I previously said, all of the growers in New Zealand are pruning their redwoods. So that concludes my presentation. Thank you for listening.